to the set standards. And this could actually mean that the residents of Nairobi are basically not benefiting from the food fortification program, but basically the government intended to help in terms of increasing micronutrient intake for the different populations that are eating these foods. And this would actually call for further analysis of the possible barriers that are actually making people not to comply, despite the fact that a lot of investment has been done by both the government and the private sector in trying to capacity build the industry, and even in trying to create a conducive environment to ensure that the food industry is able to manufacture what is acceptable to the population. So as a way of acknowledgement, I would like to acknowledge the Ministry of Health because of the use of the laboratories at the National Public Health Laboratories for doing some of the analysis, and also the Ministry of Health for spearheading the food fortification program. Then acknowledge the university, Jomo Kenyatta University, for also availing the lab for conducting some of the analysis and even for the guidance in the work. And last but not least, the European Union for funding this particular project. Thank you, and back to you, Chair.
Thank you very much, Francis, for the presentation on food fortification, looking at compliance with the milling meal providers or manufacturers are complying to the required standards. Before we get into the question and answer section, there is an evaluation form of this session. So Jeremiah has posted the link in the chat box. We kindly ask if you can complete that questionnaire. We also need some feedback from participants. Thank you. Then on to the questions. So the question reads, what would you say in regard to the consumers of self-milled maize meal? Remember, we're talking about compliance, and I want to believe your sample size was on the commercial maize meal, but the question now is saying, what should you say in regard to the consumers of self-milled maize meal?
Thank you, Chair, and thank you, Catherine, for your question. There are many things that we can say about people who mill their own maize. One, if you look at the fortification that is being promoted, it's basically to try and increase the consumption of the macronutrients. So if we're able to get them from the foods, then even things like uptake and even bioavailability of the elements in the fortified maize flour would be high. But if people are milling their own maize, which is the general practice, especially in the rural parts of the country, would not say they'll get as much benefit as if they consumed the fortified meals in terms of the minerals, because of the minerals and the vitamins. So that is basically our major focus. Of course, they'll get the whole meal, which is also good for health, but in terms of minerals and the vitamins bioavailability, it may be basically low, because if you look at the meal, the whole meal, you also have the other components of the meal that has got things like phytates that basically bind the iron that you're interested in. So it's good to consume them in terms of getting the whole meal, but in terms of availability of the minerals and the vitamins, it may be slightly lower compared to the commercially milled maize meal.
All right, thank you, Francis, for the response. The other question from another question from Irene now. It says, why is it easier to comply to vitamin B than vitamin A and minerals? Because I think in the presentation you were saying that you find the zinc, iron compliance was very low as compared to vitamin B.
Thank you, Irene, for your question. When I was making the slide on minerals, you saw that the compliance for iron was higher compared to zinc. Yet the fortificant is supposed to be like a unit. It contains the iron, it contains the zinc, it contains the vitamin A, and it contains the B vitamins. So further work would need to be done, especially in terms of analyzing the premix, because basically we are looking at the finished product, which is the maize meal. So we'd also need to look at the premix and confirm that the premix is okay. If the premix is okay, then we need to look for another area to be able to make a conclusive conclusion. As of now, I cannot say why B is complying and vitamin A and the minerals are not complying until we check on the premix and be sure that the premix has been met to the required standard as per the specification that have been given by the Kenya Bureau of Statistics.
Thank you, Francis, for that response. Mine, I think maybe might be just a point for discussion. One thing I have noticed also, fortification in milling, some might argue it might be for those who normally rely on the commercial milling or commercial products. But to cater for maybe the rural population or for those who can't afford the commercial food products, it might. What do you think on biofortification as also a way to cater for those who can't afford the commercial or industrial fortification as a strategy to also cater for those in the rural areas, the biofortification?
Thank you, Leslie, for your comments. I think there is no silver bullet in terms of fighting macronutrient deficiencies. So we basically need to explore all the available avenues, depending on each particular population you are looking at, and see what best suits for which type of population, so that you're basically able to cater for the entire population. Otherwise, you can't find one solution for all the problems for the population. The population is dynamic.
Okay.
Yes.
All right. Thank you very much, Francis, and thank you very much, colleagues. Thank you very much to all presenters. I think we've come to the end of the session. We are left with a few seconds. So it's left for me to thank everyone from the participants for your questions, for your contributions, your suggestions. Thank you to all the presenters. Thank you for the insights, the new knowledge, the new insights that you presented. And we wish you the best as you continue with the presentation, with your research, and we look forward to some of the results you are going to get. I thank you, sincerely thank you. Thank you also to the technical team who were working behind the scenes to make sure this session goes on well. And my sincere apologies for the technical hitch when we started. Somehow my computer decided to froze at the wrong time, and I had to switch on to the phone, etc. So I thank you very much. I think very soon we'll cross over to the main auditorium where we join everyone. So I think you have some on your screen. There's some information that the breakout room will end in the next 15 seconds. So thank you very much, colleagues.
Thank you.
Good afternoon, everyone.
Good afternoon.
Oh, Dr.
Afternoon.
So I'd like to welcome you to the afternoon session of today's conference program. We start with a plenary session where I will be the chair. And I've also seen that our guest is online. So this will go for half an hour, then we shall break out for the breakout sessions as usual. And I'd like to, at this moment, to recognize the presence of Dr. Vanna. So Dr. Vanna will be speaking to us on the topic of way forward for increasing resilience and food security in Africa through frontier agriculture. A little about Dr. Vanna is that she's a lead economist in the Africa region in the agriculture and food practice of the World Bank. So she leads the food security agenda. Previously, Dr. Vanna worked as a principal economist in the Inter-American Development Bank, where she led the global evaluation of IDB's climate-related interventions for a decade. She has also served as the climate coordinator in the World Bank's Middle East and North Africa region and led the Latin America and Caribbean region's work on socioeconomic implications of climate change. Lotte has published extensively in peer-reviewed journals and produced numerous books and reports on climate change, labor markets, and poverty reduction, for example. She has also developed and supervised the implementation of various World Bank projects and won 2 World Bank Green Awards and a Sustainable Development Award for her projects and research. Before joining the World Bank, Lotte worked in the Development Center of the OECD, that is Organization for Economic Cooperation and Development, as a researcher and also as a researcher at the European University Institute and Paris One Sorbonne, a leading university in Paris, and in the private sector in Sao Paulo, Brazil. Moreover, she serves in the UNHCR's Advisory Board for Livelihoods and is a fellow of the Explorers Club based on her climate change adaptation work and an award-winning photographer. I strongly suggest that you actually visit her site where there are excellent pictures showing the places she has visited all over the world. So Dr. Vanna, please welcome and let us hear your presentation. Thank you.
Thank you very much for your kind introduction, and let me share the presentation. I trust that you can see it now.
Yes, we can.
Okay, so let me just put it in presenter mode. Excellent. I don't know how fast my voice and the slides travel, so there might sometimes be a little delay. So just interrupt me if I do need to do anything differently.
Will do.
So, as Patrick mentioned, I'll be talking about food security in Africa. But I'll also talk about something that your university is a world champion on the Africa continent, namely insect farming in a circular economy model. And we do it in a way so we focus on increasing food security and reducing hunger. So what if I told you that it's possible to feed all people on the planet a nutrient-rich meals without running down the planet? That means without running down the environment, cutting down the forest, and killing the biodiversity. And what if I told that it's possible even to feed the 10 billion people that is projected to inhabit our planet by 2050? You might be saying that I'm dreaming, but I see untapped potential for producing nutritious, rich food that can improve the current food security situation in the world, including in Africa, and improve the food system globally and in Africa by applying a circular economy concept while producing. This circular economy concept will not take up more arable land, and it would not use enormous amount of water that traditional agriculture does across the planet, and it will create jobs and livelihoods for people. And at the same time, it's climate smart. It will reduce greenhouse gas emissions, create climate resilience for the farmers that produce and up the supply chain, and it would improve soil health on the continent by turning organic waste into feed. Food system in Africa already face signs of long-term challenges, including food security challenges before COVID-19 hit the continent. As you can see from this graph on the left side, since 2014, per capita food production has been falling on the continent. And in the beginning of 2021, around 240 million people in Africa experience hunger, and more so in the countries that are affected by conflict and violence. The graph on the right shows that 29% of the people in countries affected by conflict and violence experience insufficient food consumption, while in Southern Africa, no, in Sub-Saharan Africa as a whole, the number is 18%. And that is one person too many already. Hunger is not the same in all the fragile countries. We see that in Niger, South Sudan, Burkina Faso, and Mali, more than 50% of the population, more than half of the people living in these countries are acute food insecure. And food security is actually getting worse as I speak. In more than 85% of the most food insecure countries in Sub-Saharan Africa. Those are in the so-called IPC phase 3 and more need food assistance. And it's severe or greater than in the past 5 years. That means in the past 5-year average. Projected food assistance needs through July this year are most severe in South Sudan, in Nigeria. South Sudan is likely to go into outright famine by then. Nigeria is likely to be in emergency with a risk of famine. And if that wasn't bad enough, if we look into the nearer future in the next decade, so 10 years ahead, as you can see from this graph, food security will deteriorate way beyond COVID, because this graph is not even taking COVID into account. And we can see that East Africa and Southern Africa, where Kenya is located, is worse hit than the average on the Africa continent. So that means East and Southern Africa is likely to be worse hit than West and Central Africa, the blue line in this graph. So what is driving the food insecurity in Africa? There's a number of structural drivers, and let's focus on 5. And these 5 are for long-term food security and beyond COVID-19. These drivers are the climate, it's the conflict and fragility in some of the countries, and then there is the zodiac diseases, where COVID is one of them, macroeconomic situations and fragilities, and the policies that are being implemented in the countries, both related to food security, but also related to other situations. So in terms of climate, in sub-Saharan Africa, the frequency of the climate shock has increased in the last decades. It increased from once every 12.5 years to once every 2.5 years. So these shocks we're talking about is drought and floods, hurricane like Cyclone Idai in Southern Africa, etc. The second one we talk about is the violence and conflict. And violence and conflict have also surged on the continent since 2010, with food insecurity both being a consequence and a cause of the conflict. And we also know that the zodiac diseases are increasing. The continent have experienced SARS, MERS, and Ebola. And the macroeconomic crisis, we all have seen exchange rate depreciations occurring in many countries on the continent. The foreign debt is increasing in many countries, and so are the fiscal deficits. And we see fallen GDP and job creation. So cutting across these areas is important to better align policies, and that is both food policies, agriculture policies, and other public spending intervention in general to deliver and improve food security situation and therefore better food security outcomes on the continent. And I think that new disruptive technologies are called for to increase food security, in addition to the better policies. New disruptive technologies are called for, as I mentioned, but we cannot do it in the traditional way and run down the planet and the resources on the planet. I'm talking about the frontier agriculture as such disruptive technologies. What are frontier agriculture? It is the kind of technologies many have existed for a while that use much less arable land, much less water, and that really produce nutritious food, ideally at a high speed. I'll get more into the details of what I'm talking about later in the presentation. So we need to move away from a production method that is linear. And that is one that you can see that is based on take, make, use, dispose, and pollute. And we need to move more towards a circle where we make, reuse, reuse, remake, and recycle. And therefore, there's not any waste ideally or very little. And in this circular economy, we can create climate resilient livelihoods, jobs, and we can create income on the farm and up the value chain for people involved in the sector. And we reduce the agricultural footprint on the planet. As you might know, is that at least 25% of the greenhouse gas emissions on the planet come from agriculture. And we're not even taking food in that. This is just straight out agriculture. And so the agricultural footprint is not only climate change, it's also the soil degradation that we're also seeing in many places, including in Africa. We see deforestation and biodiversity depletion. We need really to include food security, as I told you before. And we need to expand the menu of foods that are available and that are little resource intensive. I've had the privilege to share protein-rich meals with people across the globe, including in remote areas in many countries. And in Zimbabwe, the Zimbabwean taught me that a crunchy mupama worm is a good meal. 90% of Zimbabwean consume insects. In South Korea, the South Koreans taught me that mealworm can be used to spice up salads and make desserts delicious. There can be ingredients both in desserts, but also in drinks. The Indians, they shared me their secrets when we cooked red weaver ants for the dinner, and we enjoyed a meal of ant sauce and rice. It was delicious. Ants are hard to catch, but trust me, to prevent them from running out of the pot, it's even harder. Food connects us, and we all like to share a meal. There are probably 2 million people, according to FAO, that collect and eat insects as part of the diet on the planet. And what we have seen in many places, including in Harare in Zimbabwe, there's a market for insects, as this picture shows. As a matter of fact, insects now in Southern Africa are traded across borders. So Mopama worms are harvested in Zimbabwe and sold in South Africa and in Zambia. Insects are nutritious and delicious, like any other well-prepared dish containing healthy ingredients. Insects have many of the good amino acids that we need, and many of them have all 9. Many has iron, zinc, and calcium, and some of even have omega-3. These are all some of the micronutrients that children in Africa are lacking in many places. There are challenges with collecting insects, including both the collection and the safety in consuming collected insects. First, most of the collected insects are collected in the wild, in the nature, in specific period of the year. That means they're seasonal and not available all the time. Mopama worm, for example, can be collected around 4 months per year. Also, insects, they eat what they feel like. So they would eat of trees and crops, and they don't distinguish if these trees and crops actually have been sprayed by pesticides. So the food that they eat is contaminated with dangerous chemicals for human health. For example, one of the women I talked to that producing in Mexico, she told me that many of the grillos that are sold in the markets in Mexico, they are actually caught in the wild on corn, and they are feeding off corn that have been newly sprayed, so they're contaminated. So what if we stopped over harvesting the insects in the wild and we supported the ecosystem by producing the insects, by farming insects instead of collecting them? Then we would solve these 2 problems I just mentioned. We would have a year-round supply, no seasonality in supply, and we could control what the insect eats. So no dangerous chemicals will be moving up the food chain. We can raise insects like we raise other livestock. And I would like to give a shout out to John from your university, that is the leader in this in Africa. John is doing research on crickets and other things, and is doing pilots in different places, and really have been working a lot with us in this project, because it's the researchers in Africa that sit with the knowledge. Us in the World Bank, we're trying to learn and figure out how to do this. And that's why we're producing a report now that John, he also has contributed to. So thank you for that. So as I said, we can raise insects like we raise other livestock. Clearly, cricket and mealworm can be categorized as a micro livestock, right? We farm globally less than 20 animals. If we could increase this number by 50%, that is maybe 10% out of the 2,100 edible insects that exist. So as I mentioned, we are working in Africa, and this is the work that we're doing with John on insect farming on the continent. And we have been collecting data in 13 countries. And one of the models that we have been looking into that is very well advanced is the South Korea model. In South Korea, there already exist more than 3,000 insect farmers that produces insects for food and for feed, so human food and animal feed, but they also farm for medicine and health products, including creams and other products. Then we're working with the Rural Development Agency in South Korea on this task. As I mentioned, we're collecting data on the Africa continent, and we're working with African researchers such as John to learn where insects are actually farmed already and what kind of farms we exist and what is being farmed. So we did a farm level survey. We also did a country level survey. This is not representative in any academic way, but the findings of the survey identify 849 insect farms of various sizes already exist on the continent. The surveyors actually estimate that it's closer to 1,800 farms that exist on the continent, and it's rapidly expanding. Frontier agriculture, insect farming can benefit all ages and both men and women. The findings from our research show that 12% of insect farmers are under 30 years old and 22% are over 60. And we see each year the number of entrants of farmers and companies that work up the supply chain increases. And that is not only in Africa, but that's across the globe. As a matter of fact, Barclays Investment Bank have estimated that globally, the market for insects as human food and animal feed will be worth about $8 billion by 2030. That's an average growth rate, annual growth rate of 24%. Not many other sectors, at least the ones that I work with, can count on such a fast growth rate. So, okay, maybe not everybody wants to eat a mealworm burger like we had in South Korea. So what if we fed insects to animals? So the animals that people eat, such as the chicken, the fish, pigs, are fed their favorite food. We all know that when chicken roams in the wild, including at my grandmom's farm, the chickens, they go and they'll pick the insect rather than anything else. Yes, insects or feed with insect protein as the main protein source instead of fish meal or soy meal. Today, we are feeding fish. About a quarter of all global fish catch, so the fish that are caught in the sea, goes to produce meal that is fed to farmed fish. So the salmon, the tilapia, etc., that we have globally. That is a waste of the fish that is caught in the wild, and it's also highly unsustainable. So we could also reduce the soybean, not only the fish caught in the wild, but we also could reduce the soybean that is put into animal feed and fed to animals. With that, we could maybe stop cutting down the rainforest and other tropical forests, including in South America. As we know that, for example, the Amazonian rainforest has been cut down first to produce cattle, and then it's basically to produce soybean. Today, 70% of the planet is used to feeding livestock. That means 70% of the planet, we grow crops that we feed to animals. But we could produce that feed, including on Africa. And I honestly believe that Africa could be the global leader in insect farming. So what does an insect farm for feed look like? This picture shows an example from South Korea that is producing black soldier fly larvae. How long does it take to raise a chicken or produce soybean? I'm not sure, and that really depends on the place and the environment, but I know that black soldier fly larvae can be produced in 2 weeks in limited space with no arable land and very little water use, and it's drought resilient. So it's a highly favorable thing to farm in many places in the world, including in Africa. So let's come back to how we suggest ideally to produce insects, and particularly when we do it at a large scale. And we should suggest to use this circular economy model here. So this is an example, this is a draft, the sketch, how we can maybe do that. So let's start by producing insects with this circular economy model. So we have to ask the first question, what does insect eat? Well, insect can be fed organic waste, household waste, agriculture and manufacturing waste. So that's crop. When we have, for example, taken the corn off the cob, we have part of the crop that is left that usually will be burned or maybe fed to cattle or just left to rot in the field. And we also know that manufacturing sectors such as breweries produce waste. So this brewery waste we can also feed to the insect. So insect farming can actually reduce and help reduce the waste that countries face that usually they have to pay to get rid of, right? So not only we can feed, for example, the black soldier fly, these waste products, but black soldier flies, they have a voracious appetite and they can produce a lot of protein, and in just 1 square meter of black soldier fly, so a small farm of 1 square meter, they can produce about 15 kilos of organic waste per day. So the insects are consuming the waste that we throw away or burn in the fields that contain greenhouse gases. Therefore, insects actually can contribute to reduce climate change and solve our waste problem by turning waste from a liability in the economy into an asset. So in this process, we produce insect larvae that can be turned into protein, to oil, and to frass. And this frass can be turned into a biofertilizer. The protein will be fed, for example, to fish, to chicken, to pigs, that serve as food for people, and that create incomes and jobs along the way. The people will then produce more organic waste that will be served to insects. So this is a circular economy that I'm talking about. So what did we learn from our survey? We learned that nearly half, 43% of the survey insect farmers use household waste as insect feed in Africa. And 64% use industrial food waste, mainly brewery waste. 12% of farmers use manure. And many, of course, use a mixture depending on availability of the different substrates. So this makes black soldier fly a good candidate for breeding in countries where organic waste management is a challenge. And that's especially in cities. So where are we thinking of starting to move ahead and support insect farming? We're looking into very food insecure countries and countries that face hunger, and at the same time that have difficulties in generally getting started. So the fragile conflict, violent countries such as South Sudan, Zimbabwe are some of them. So if we collect the agricultural waste and feed this to the insect larvae, to the insect to produce protein, to feed animals, how many jobs, how much greenhouse gas emission, and how can the soil health be produced? We did some calculations that are clearly rough, but we took the top 5 crops in 10 countries. And I'm just going to give you one example here of Zimbabwe. We took the top 5 crops, the maize, the groundnut, the soybean, the wheat, and the sugarcane. And if we hypothetically assume that we could collect 30% of that, I mean, I'm talking about the waste, right, from these 5 crops. And we could collect 30% of this waste and feed this agricultural waste to the black soldier fly. Then we could produce up to 860,000 tons of wet larvae. And if we take the value that we are hearing that the wet larvae has in Africa right now, that is a value of $38 million in Zimbabwe, basically by using waste. In Zimbabwe, they farm already around 27 million animals, and that is fish, chicken, goats, and pigs. And that require around 700,000 tons of protein per year. So with a waste-based protein from black soldier fly larvae, that could meet 20% of the annual protein demand in Zimbabwe. And how many jobs will we create along the way? By using 30% of the waste, of the agricultural waste, we calculate that we can create about 60,000 direct jobs and 160,000 indirect jobs. So that's more than 200,000 new jobs, pretty much in a heartbeat. In addition to these social economic benefits, there are also the environment benefits. By not burning or leaving these these agricultural waste products to rot, we also reduce greenhouse gas emission. Greenhouse gases from 1 ton of organic waste in a landfill produce 1,700 kilos of CO2 equivalent, while black soldier flies produce 35 kilos of greenhouse gas, the CO2 equivalent. This is huge. And by this, we also protecting the rainforest, as I mentioned, by not cutting down the rainforest to produce more soy beans to put in feed to feed animals. And as I mentioned, these black soldier flies have ferocious appetite. So all the frass can be turned into organic fertilizer and fed to the soil and improve the soil health in Zimbabwe. So what are the ways forward? So we did this analysis not only for Zimbabwe, as I mentioned, but for 10 countries. And I'm not going to get into the big numbers. You can see it in the report when it gets released in a few months. It is huge, absolutely huge. So the way forward. So how do we actually go from this more theoretical way of doing this to implement this kind of farming, frontier agriculture, insect farming on the African continent and beyond, really? The way that we are seeing it as of now, this is work in progress, as I mentioned. We are focusing on 2 phases. So we have to establish the sector. And we have a lot of evidence from what is already existing, including in Kenya, from all John's and his Zippers and others' work. And parallel by establishing the sector, including getting the regulations in place and in the place that we want to produce, because not every country has regulation that allows for insect farming. So parallel, when we establish the sector, we start to pilot and working with research, the way that John he has done so effectively in Kenya when he has been implementing cricket farming in Kenya and beyond. So when that phase is established, if you will, then we can move to scaling up and propel this circular economy throughout. And as you can see from the slide, there's different steps, but let me stop here and just say that insect farming is a huge potential. We can, if we go for a linear model to a circular food production model, we can really, really change a lot of things on the African continent. We can improve the food system and feed more people and reduce malnutrition. We can recreate climate resilience livelihoods, jobs and income from the farm and up the value chain. We can contribute to reduce the agricultural footprint on the planet, from soil degradation, climate change, deforestation, and biodiversity depletion. And at the same time, we can save hard currency by producing protein instead of importing them. This is sort of the macroeconomic impact that I didn't talk a lot about, but it's also huge, particularly for the cash-stricken countries. And if it's possible to feed all people on the planet protein using a circular economy model, I'm not saying that all humans need to consume insects. Insects can be fed to the livestock and to the fish, as I mentioned. But I just will share with you, if you have never eaten a dessert made of bee larvae and white chocolate, you have missed out. It's amazing. Thank you very much.
Thank you very much, Dr. Vanna, for your very interesting presentation on frontier agriculture. I think many of us were expecting something to do with irrigation or better climate smart agriculture, but this is interesting because even in Kenya, some of our communities do actually eat insects as part of their seasonal meals, and they prefer it far much more to the conventional meals that we have. And yes, Dr. Kinyuru has been very instrumental in pushing us to actually even accept to eat crickets, especially some of us who are not from the insect-eating tribes. So I will now open up the discussion for a few questions for Dr. Vanna. Please remember that it is quite early in the US. She's just woken up, so as the questions come, we could take a cup of coffee, but yes. So questions, welcome. Questions, observations?
So check on the chat. Check on the chat.
Yes. Thank you. Checking on the chat. So the first question is, which model is the World Bank going to use to promote insect production? So I've seen that you've talked about the circular economy model. Is that what you're looking at, Dr. Vanna?
I would say, I think the learning will be in the making, right? We know from the survey that John here also helped us work on, that there is a lot of this circular economy that is already being used. It's actually already being done, right? For example, as farmers use brewery waste and agricultural waste and household waste to feed insects. So it's not about what we see, it's about what is available in both in the community, really. It's not only in the country, but it's also in the community where these farms are and where will they pop up. So it's not, we don't see that as one model, there's many models, but this is more the way I see it, as a concept that can bring ideas. And as with everything that is new, everything that is frontier, our imagination is really what stops us, right? So many of the things that John he has shown me and taught me have really expanded my horizon. And John has been sharing his experience with farmers in Kenya, meaning their horizon has been expanded. So frankly, the sector, the insect sector on the African continent and globally really has been stopped because there's been too much private information. There's been too much focus on mechanization, like in the US and Europe, and even some places in Asia. Focus on mechanization and all this private information because it's very expensive and labor costs are high, and to compete with the price on the ground of soybeans in Africa and in the US, etc., the price has to be really low for it to be a competitive input to the feed for animals, right? And that's why Africa is amazing, because people are creative. There's a lot of ideas, and I believe there can be a model developed that we might not even know, or I do not know. Maybe it's developed in some places, and that's why I hope that when we have this report ready, that we can maybe do some Zoom, because I'm afraid that because of COVID, we still can't travel. And maybe we can do some Zooming where we discuss our findings, basically to jointly advance, because us in the World Bank, we cannot do this sitting in Washington. It has to be the people in Africa. We can help aggregate the text. We can maybe help with finance if there's a demand from the government and these things. But we can't be the one that guide the way that I've seen John, he does in Kenya. And I have seen Robert do in Zimbabwe. And there are many people on the African continents, given that we find that there's insect farming in the 13 countries where we survey, and there's countries that we didn't survey where we know there's a huge demand and people would like to get into this. And then let me stop here.
Three more questions. One is about the culture change for communities that traditionally do not eat insects. So how do we get them to accept insects as really good food for them? The second one is on supporting communities that, for example, NGOs so seed capital in conflict areas. So like you talked about Niger, you talked about other places like Sudan, where they might be having trouble in conflict. So can some local startups be given some money to do that? And then third is your take on using human waste for insect production. What's your feeling about that?
The first question, let me be very clear. The demand for protein is so huge. And it's not about, for us, for me, it's not about changing people to like it. I had never honestly eaten insects before a couple of years back. And I'm a vegetarian, okay? Now I eat insects. You don't want to hear about my story. That's not relevant. But given the demand for feed for animals is so huge, and very few people that I know, pretty much none, really look into what is the chicken that I buy, either from the farmer or the supermarket fed. Is it soybeans? Is it insects? Or is it something completely different? What is the mix of this feed, right? Some people are concerned about it's organic or not. But most people they want it maybe to be free range, that usually globally grab a higher price because they have been out in nature and maybe eaten a more diverse diet. They become more tasty, people say. So it's not about converting people. But as I say, if you don't have the opportunity, you don't have the choice to pick a biscuit, like some of my colleagues from the University of Copenhagen, Nana Rose and John, have been working on. You know, if you don't have a choice between a biscuit with a high nutritious content, a high amount of protein, and a very sugary, zero nutrition protein, when you are in the supermarket to buy biscuits for your family, then you miss out, your kids miss out, and the whole country and the continent and the world miss out, right? It's about creating choices. And at this point, there is not even enough insect produced that we could even think about of how to convert people. And this is not the point of this work. The demand is huge and the needs are even larger. So to start with, it's about feeding the demand that already exists. And then we'll see if more demand is being created. I can just say from what I'm seeing in some of the countries that really have started up and done this fast, like in South Korea, there's a huge demand, right? So we'll see, and maybe John and others have a better sense of this in Africa, including in Kenya. The second thing was, there was one about the waste. I don't have a view on that. I know that there are some experiences going on in Africa with houseflies, with manure, and I also know there's some experiences going on even in Kenya, in Nairobi, with using human waste. I don't know enough about it, and at this point, I don't feel comfortable saying what I think, because I haven't really thought about it and studied it enough. And there was one question about for NGOs. I think there's a job here for everybody. It's a job for investors, the job for governments. And the investor can provide finance, government can provide regulations, research support, training, extension service, trying to build public-private partnerships. The farmers can create, the farmers that are interested in this can request training, they can request support, they can create farmer organization, because as we know from many other fields, not from insect farming, from other agricultural fields, right? If you can aggregate, so if you reproduce eggs and you can put your eggs together with your neighbors and the communities, you can maybe get a higher price and sell to somebody that demand and sell it at a bigger scale. So there's all these things. So there's a job for everybody. And NGO, in many other ways, have had a crucial role in starting things and being the ones that are providing training and providing the resource and linking maybe farmers with governments. And so there's a huge role for NGOs. As I mentioned, that we are planning to do some small pilots in South Sudan and Zimbabwe, Malawi, and maybe in other places if funds become available. For this kind of work, we also have to go out and fundraise, like NGOs has to fundraise. But if you have an NGO that is working in India's countries and you're already doing it, feel free to reach out to some of the researchers or even send me an email, because I would like to know about this. John is one of the most incredible leaders in this on the African continent. He's probably one of the ones that have the biggest Rolodex of contacts about people that do produce. So, and I hope as we move forward that this work will really take economy-wide interest So that means that it's people in communities, rural and urban, it's young people, it's old people, it's middle-aged people, it's women, it's men, and it's community organization, it's NGO, it's private sector, and it's government, both the local and the national. And I think I'll stop here.
I think we'll also stop there so that we give you time to prepare for your other activities. And we really, really, really want to thank you for finding time, waking up early to be able to give us this very interesting view on what we have seen John do in the university. And now you've actually blown our mind and told us that it can actually be not only a Kenyan, but a regional and even continental-wide initiative. So I will stop there and hand over to Dr. John Kinuru, but also thank you very much for making time to give us this very interesting talk. Thank you, Dr.
Thank you for the opportunity.
Thank you very much, Chair, and thank you very much, Dorte, for this brilliant presentation. There is, however, a question by Fokwa Fombo from Belgium. Can we have her email address? Dorte, do you want to give people your email address?
Yeah, that's fine. I will write it in the chat now.
Beautiful. Thank you so much. Thank you, and have a great day ahead. We appreciate your presentation. Thank you, members. Now we move on to the plenary, to the parallel sessions. I will be opening the breakout sessions now in a minute. Just like in the morning, kindly pick the breakout session that suits you. We will have 4 plenary sessions that will go on from here. We'll have a session on information and communication technology for development. We'll have a session on agricultural sciences and technologies for sustainable food and nutrition security. We'll have a session for policy, governance, entrepreneurship, cultural and socioeconomic aspects for competitive advantage. And we'll have a conference exhibition going on. So those 4 sessions will be open for joining. Kindly do not join the other sessions that are not being offered, but those 4 sessions will be ready. Thank you. Click out the breakout room on your bottom right of your screen and move on to
Good afternoon, everyone. I want to take this opportunity to welcome you to this breakout session. I'm waiting for the rapporteur to join us so that we can proceed. William, if you're here. So good afternoon once again. I want to take this opportunity to welcome you to this breakout session. In this session, we're going to have... Hello?
Yes, Doc, we can hear you very well.
Okay, thank you. Jeremiah, you'll be the rapporteur?
I will.
Okay, thank you. Thank you, Caroline. I was expecting William, that's why I was waiting. So thank you so much for joining us in this session. In this session, we're expecting 6 presenters who will be presenting on different topics. The first presenter, and this will be the order of the presentation. The first presenter will be Diana Debra. The second presenter will be Agnes M. Kavo. The third presenter will be Kones Cheruiyot, followed by Clement Komu, followed by Elias Ngotho, and finally Victor, I think there must be Gikunda or Victor Chege. So that will be the order of the presentation. Just to remind you, the presenters, you'll be expected to display your slides, and you'll move your slides the way you would prefer to present. The second thing is that you have 10 minutes for this presentation. When you get to around 9 minutes, we'll remind you that the time is up. We'll give you an extra 5 minutes for question time. I would request those of us who are in this session, if you have any question for the presenter, kindly jot it down in the chat so that we can be able to all access it. And I will take time at the end of the presentation to read out the question to the presenter. In case you have a burning question that you would want to air, of which we highly discourage, you can raise up your hand. When I see your hand raised, I will unmute you and you can be able to ask the question to the presenter. So having said that, I would call upon the first presenter to start off with the first presentation. And that should be Diana Debra. So Diana is going to be presenting a paper. Her presentation is titled Effect of Intra and Intergeneric Grafting on Physical and Biochemical Properties, Post-Harvest Physiology and Sensory Attributes of Tomato Fruit. Welcome, Diana.
Yeah, let me share my screen.
Diana, do you need any assistance? Okay.
Diana, do you have a problem sharing your screen? We can share it from this side.
Hello, Diana. If Diana is Diana, if you're facing any technical problems, we can assist you. We can open the presentation and you can present as we assist you to move the slides.
Excuse me. She says that
her laptop is a bit slow and it's not working right now.
Share on her behalf.
We're not in the same place. She just texted me.
Okay. In that case, then we'll call upon the second presenter. As you kindly, Margaret, assist her to sort that problem if you can be able to access her. In the meantime, then we can call on the second presenter. The second presenter is Agnes Presentation. She's going to be presenting on farmer knowledge and preference for orange fleshed sweet potato production and utilization in Isiolo County of Kenya. Dr. Agnes. Jeremiah, do you have her presentation?
Doctor, that one I didn't have.
She says she sent it around 2:30.
Oh. The name is
Hello?
Hello.
Is someone?
Hello.
Yes, Dr. Agnes.
Yes, Doc. I sent a presentation around 2:30.
Yes.
Someone sent her presentation out to that. She's asking for it.
Maybe as we wait for you, Dr. to sort yourself out, we could ask if Conness is ready. Then you'll present next.
You have his presentation.
Jeremiah, do you have Conness's presentation?
Hello, Chair.
Yes.
I think we have a problem with those presentations that were sent just a while ago because we had collected all the presentation from yesterday and the days before and even this morning.
Yes.
So I think those people who are ready to present, and if you send your presentation today, please let us know so that we can be able to pull it out from the mailing list. Then whoever is ready can present first.
Okay, so thank you so much. So Conness Chariots, are you ready?
Doc, yes, my presentation is ready.
Yes.
You have it there. You can upload.
Okay, I can see somebody sharing. Yes, that is the one.
This is for Agnes.
Oh, okay. Then that takes us back to Agnes' presentation. So Agnes, like I said, is going to be presenting to us on the farmer knowledge and preference for orange flesh sweet potato production and utilization in Isiolo County of Kenya. Welcome, Agnes. You have 10 minutes for the presentation and an extra 5 minutes for questions.
Okay, thank you, Doc, for the opportunity. I am presenting on the farmer's knowledge and preference for orange fleshed sweet potato production and utilization in Isiolo County. On behalf of the project partners, Dr. Mwashasha, Dr. Kariuki, and Dr. Kirika. As a way of introduction, undernutrition and malnutrition are leading challenges for many communities in the world today. And it is estimated that 870 million people in the world are undernourished. And among those, 852 million live in the developing countries. And this problem of malnutrition is mostly unique to children and expectant women. And in the arid and semi-arid areas of Kenya, this is a major challenge affecting children and women in those communities. This problem of micronutrient deficiency can be strategically mitigated by supplementing dietary intake of micronutrients such as vitamin A. And the orange fleshed sweet potato has been found to be rich in beta carotene, which is the precursor for vitamin A. And this can be very instrumental in alleviating the nutrient deficiencies, especially among the communities living in the arid and semi-arid areas of Kenya. Next slide. The sweet potato production has been identified as an important crop in the world and in the sub-Saharan Africa. And in Kenya, it is currently a very important crop, covering about 4% of the total area under cultivation. And it is mainly practiced by small-scale farmers who are concentrated in the arid and semi-arid areas. And it is important as a food security crop, mainly due to its short maturity period and the fact that it can be harvested as a piecemeal food. So that means that the farmers can enjoy the harvest over a prolonged period of time. Next. So for this project, the main objective was to document the constraints of orange fleshed sweet potato production and to assess the potential for adoption and if possible, develop mitigating measures for the constraints so as to improve food and nutritional security among the Astral communities of Kenya. Specifically, we assess the farmers' knowledge and production practices, constraints, acceptance, and willingness to grow the orange-fleshed sweet potato. This study was conducted in Isiolo County in 4 areas, Burati, Garemara, and Bulapesa, and Isiolo Central. We used a participatory rural appraisal approach where we purposely sampled the farmers. We interviewed 150 farmers and using structured questionnaires and focus group discussions. And we integrated the questionnaires into an open data kit. We collected information on the social and demographic characteristics of the farmers' households, the production systems, the agronomic practices, the quantity, the harvests, and storage and utilization of of sweet potato, and specifically the orange-fleshed sweet potato. Then we analyzed data using the SPSS and reported using descriptive statistics. So from our findings, we found that quite a high number of male were actually growing sweet potato, and this is an important aspect for assessing adoption, because if the household heads participate in production of sweet potato, then it's a plus for making decisions on the adoption. And we found that the crop farming was contributed to the highest source of income. And being mainly a livestock producing area, we see a shift into adoption of crop farming, which is important for for deciding on adoption or introduction of the orange sweet potato within that area. The production systems, we found that sweet potato farmers or production was mainly done for about next slide, for about 1 to 4 years on average. That was the height on aggregates. Most households had practiced sweet potato production between 1 and 4 years. Next slide. The sweet potato varieties that were mostly cultivated were the red skin, white flesh, and the white skin, white flesh, white skin, orange flesh was also cultivated by the communities. And the reason for cultivation varied among the household or the respondents. We see the orange sweet potato, or the orange fleshed sweet potato was most preferred because of its palatability, which is a good thing for adoption. And even the yellow flesh, cream skin was also highly palatable. And the local varieties were also highly palatable, but the most palatable was the orange fleshed sweet potato. The next slide was the sources of seed. The farmers mainly used recycled seed. Some used improved varieties, but the level of seed cycling was quite high in these communities. The next slide, harvesting and storage. We found that most of the farmers did not store the sweet potatoes after harvest, meaning that they were practicing the piecemeal harvesting. And this is important because it means that farmers can have food for a prolonged period of time, because as we know that the sweet potato has a short shelf life. Then the time that the piecemeal harvesting farmers took to actually consume everything that they had planted in the field, we found on average that most respondents or most of the households were able to harvest or consume the sweet potato from the farm for at least 3 to 4 weeks. A number of constraints, next slide, were identified by the households. Yes, and as you can see, there are quite a number of major constraints that were identified by the farmers, including access to extension services and a poor, lack of government participation and poor storability or inability to process the sweet potato into a storable form were identified as the greatest constraints to sweet potato production. Sweet potato utilization. Next slide.
And you have 1 minute to go, please.
Yes, we found that the households were able most households use the sweet potato as it is, either as boiled, roasted, or baked, but most of them did not process the sweet potato into any other form. About 52.4% of the farmers were aware of the improved varieties and were willing, 100% of all the interviewed households were willing to adopt the improved OSP, the orange-fleshed sweet potato varieties. That is, and in conclusion, please move to the next one. In conclusion, we found that about 50% of the total household income was actually from crop farming. And as I said before, this is a good indicator of a shift from the traditional pastoral community to a more settled agricultural community. And it is a good entry for introducing the orange-fleshed sweet potato production in this area. The study also revealed an existing potential for adoption of the sweet potato, with a willing 100% of households willing to adopt it, the orange sweet potato, because of its early maturity and being the most palatable. There was also a potential for multiple utilization of sweet potato and its products, and this is important for scaling up and adoption or production. And there was various constraints that we identified during the study, and that report is key in informing policy or devising capacity for developing tools for apps.
Okay, thank you. Thank you so much for the very nice presentation and very clear presentation. I want to take this opportunity to request anyone who could have a question for Agnes and team to put the question on the chat.
Yes.
Any question on the chat? In the meantime, Agnes.
Yes.
Can you hear me?
Yes, I can hear you.
Yes, I have one question as we wait to see whether we get any other questions.
Yes.
One of the social scientists have time and again told us that wherever you see the male population interested in the production of a certain crop, it means that they are looking at the commercial aspects of the production, not probably the nutritional aspects of the production. What was your experience with this community? Was it that they are growing the sweet potatoes for commercialization, or are they growing them for domestic consumption?
Most of them, thank you, Dr. for that question. We found that most of them were growing for food because this crop was introduced by other partners, some NGOs, initially for food security, food and nutritional security. So what we found is that they mostly used it for consumption, for food, but they were willing to grow more so that they can commercialize. They can grow for commercial purposes. And as you can see that already they were using the crops, some other crops for income, for household incomes. So there is potential for commercialization of this crop. Thank you.
Thank you. Thank you so much for that very, very clear answer. And we seem not to have any other question for now, but just in case we get any other questions, we're going to direct them to you. So at this particular time, I want to request if Diana, you are ready now. Diana?
She's not.
She's not. So then we can go to the person who's supposed to be the third presenter. Leonard, I understand you said you are ready.
Yes.
Oh, but I can see something open for Diana.
Okay.
So Diana?
I'm sharing from this end. I don't know whether she is ready to present. Maybe if she is not ready, I can just open.
Yes, stop sharing. Let's have the Leonard, because he's ready to continue with the next presentation. The next presentation is on isolation and characterization of rhizospheric microorganisms from bacterial wilt endemic areas in Kenya.
Yes, Chair. Do you have the presentation loaded? Because from that end,
We can assist Jeremiah.
Yeah.
Okay, Leonard, there you go. I think the presentation is ready.
so good late afternoon, everyone. My name is Leonard Kireka. I'm here presenting in place of one of our students, this project called JKUAT, and this project is part of a collaborative project involving several other scientists like Dr. Mwashasha Rashin, Dr. Lilian Kariuki, and Dr. Agnes Kabo, who just presented. So I will go direct to the presentation. So in this section of the research, we were screening of beneficial rhizospheric bacteria and fungal isolates for control of bacteria wilt of tomato in Kenya. So I go to the next slide, which is the introduction. So tomato accounts for an equivalent of 63 billion US dollar in a year. And it contributes to about 26% of the annual GDP indirectly, and 25% indirectly, was study done by Maora in 2013. And the production of tomato has been limited by challenges, which include pests and diseases. And you can see there on the left side of the slide number 2, the production statistics there, the bar graphs. And on the second bar is where you have the tomatoes related to other crops.
Sorry, Doctor, we can only see the first slide. Jeremy?
Yeah, please go to the next slide. Are you able to go to the next slide from that hand? Okay. All right, there you go. Oh, so, thank you. So on the second slide, just as a repeat, I've introduced the tomato, and I've seen that it accounts for 63 US dollar billion per year, and it contributes to about 26% of the annual GDP directly, and indirectly 25%. The production of this crop is declining, and this is due to limitations limitations due to pests and diseases. And if you see, compared to crops in different regions of the world, on the left is where I have, that is, I'm on slide number 2, please. Slide number 2, go back, please. Yes, there. So on the left, you can see a chart which has the bar graphs. On the second graph, bar is where you have the tomatoes compared to other vegetables produced. So go to slide number 3. Yes. So in this slide, it's the statement of the problem, whereby one of the major challenges in tomato production is a bacterial wilt cause organism, Ralstonia solanacearum, which is a devastating soil-borne disease, which affects tomato largely all over the world. And it has negative impacts on the plant, which has been reckoned to amount to yield loss of over 90%. And there is no single mechanism of control is already efficient that has been reported across the world. And therefore, based on this fact, there was a reason why we embarked on this research to try to find ways of effective control of these pathogens. So I go to slide number 4. Yes, so the reason why this study was conducted is because there is a rising demand for the food in the world, and also the challenges of environmental effect by use of a lot of pesticides in the environment. So there is need for use of the BCA, the biological control agents, for better food production. So in this case, we are backing on studying the rhizospheric environment of the plant that are showing tolerance to bacterial wilt. And therefore, we observe that the rhizosphere contains antagonistic microorganisms which can provide immunity to plants against diseases. And on this line number 4, you can see the biological control agents market chart by Validindi and Panda in 2019. Please go to slide number 5. Yes, so these are the objectives for this study, which the main objective was to screen for the rhizospheric beneficial bacteria and fungal isolates for control bacterial wilt of tomato in Kenya. And the first specific objective was to evaluate the beneficial rhizospheric bacteria and fungal isolates from bacterial wilt tolerant tomato plants. So we included also fungal, since it's also a challenge in tomato production in those selected regions in Kenya. And also the second was to determine the efficacy of these specific identities of microorganisms isolated from bacterial wilt regions, or bacterial wilt plants that harbor bacterial wilt in those selected regions. Slide number 6. This is a slide showing a pictorial representation of materials and methods that are used. On the first box, you can see the student there used to sample in a tomato growing region in the selected counties of Bomet, Kirinyaga, Kajiado, and Kiambu counties. And these are the counties that are identified to produce tomato. So the initial section was to identify part of the field whereby the plant is showing tolerance to bacterial health, and whereby the plant was sampled, and also the soil was sampled, and the samples were stored at 4 degrees Celsius, which were then, after storage, they were obtained and prepared and spread on a PDA plates, incubated at 28 degrees Celsius for 72 hours. And after that, were purified and identified according to Burnett and Hunter. And also the plant samples were, I'm on slide 6, please. Yes. So similarly, the samples, the addition of extracts from the plant samples were plated on plates, and then identifications were done. So now slide number 7, please. This is for objective number 2. We are also doing the pathogenicity tests. This involved isolation of Ralstonia, whereby tomatoes were then inoculated with these isolates to observe the symptoms. Then in vitro assays were done to determine the efficacy of isolated fungal and bacteria from these regions. So this is on the right, you see the methodological flow charts of the methods that are conducted. The next slide, please. In the next slide, you can see the identities of bacteria and fungal structures from different regions, from Kiambu, Kajiado, Kirinyaga, Bomet. And as you compare between the bacteria and fungal, you can see the identities there. Like, for example, in Kiambu, we had 12. And for fungi, bacteria, we had 11. And the highest for the bacteria was identified from Bomet. And that is on table 1. Then table 2, you can see the coronary and microscopic characteristics of the identities of the fungal structures. And they were also identified, you see on the right side, remember we have the microbial species that are identified. And you can see we had observed Trichoderma species, Aspergillus species, Fusarium, Phytophthora, Penicillium, etc., etc. So these are the identities of the microorganisms that were identified in those regions. Slide number 9. This slide shows the colony and the microscopic characteristics of the identities of the fungal isolates. And you can see on the left column is the total number of isolates and the different characteristics. So there was observations down on the surface, on the rivers, on periphery, the margin and the hyphae, and then the identity given the name on the right. So you can see different isolates on the right far hand row, which has the Trichoderma species all the way to Ondecaphlorum species. And at the bottom, you can see the microbial identities, morphological characteristics on the plates for A and B and B. For A, you can see the whitish green with smooth airy hinge, spherical, vesicle with the conidia rising from the vesicle, and then the last one is the sickle-shaped microconidia. So these are the characteristics as identified on the in vitro on the plate. Number 10, slide number 10, please.
Your time is almost up.
Yes, I'll try to finish up. So these are also the other identities for bacteria isolates on the first table. And the second table, that is table number 4. These are the isolates also for the observations for the bacteria isolates that are identified in this study. And then number 11, slide number 11, this slide shows the appearance of this specific identity or individual isolates of some of the biochemical reactions. And you can see how they appear from A up to M. The results reveal that the presence of diverse isospherical mechanisms in the soil were present, and the composition of these bacterial communities then defined according to the root zone where they were identified. Slide number 12. We also did the in vitro assay, where all the 42 fungal structures and 41 bacterial solids were subjected to the in vitro assay, and some showed inhibition between 11 millimeter, which were contingent to being effective against plant pathogens. And so out of those 41, only 4 were found to be effective to inhibit the growth of bacterial wilt. And then there are also antagonists with lower molecular volatiles, which are good in controlling plant pathogens. So these are the in vitro assay, inhibition assay for the microbial interactions. Slide number 13 is the conclusion out of those studies that the plant rhizosphere, whereby the study was done showing the tolerance, consists of both beneficial and non-beneficial microorganisms, as indicated by the identities that I've just presented. And some of the rhizosphere bacteria are potentially able to cause diseases like the Ralstonia serracerum in tomatoes, and then find in those rhizospheral environment. Then these beneficial rhizospheric microorganisms have the potential to control plant diseases, as shown by the inhibition assays as conducted by in this study by this student. So in this case, we can conclusively say that in these regions whereby we have the soil microbiome, the microbial world underground, we have existing pathogens and existing beneficial microorganisms, and upon interactions with the host, they are able to convey or to subject some level of tolerance when it comes to pathogen and the plant interactions. And so this study was funded by the Africa and Japan, and it's part of a study. So thank you very much for the time and for giving me a chance to also to listen to me and in this presentation. Thank you.
Thank you so much, Leonard, and your team for the very nice presentation and very good work that you've done. we've seen you've worked with a lot of with a diverse number of microorganisms in your work. And so there's one question that has been posed by one of the members of this audience, and he would wish to understand how you identified the bacterial isolates.
Thank you very much for the question. Initially, we did morphological using morphological characteristics that we know of bacteria isolates. Obviously, for example, the bacteria wilt, we checked when you go to a slide that I have just presented, like slide number 10, whereby we have the microscopic observations on bacteria isolates. You look at the cell shape, you look at also the motility, the gram reaction, also the morphological characteristics, like you use the T's and Z, tetrazolium chlorine, which is shows the metabolic activity of the bacteria. And we know the bacteria is pink-centered colony. And yeah, so those are some of the characteristics we basically used as morphologically. But we have an opportunity of going ahead and identifying these isolates in molecular aspects. And this is something that is ongoing.
Yeah, thank you for that answer. So you're also going to carry out the molecular characterization so that you can get to clearly confirm these are the specific isolates that you're referring to.
Exactly. Yes.
Thank you for that. I want to remind all of us that you can ask any question that you have for the presenter on the chat. At the same time, if you feel that you would want to ask the question personally, you can raise up your hand. We will give you an opportunity to ask the question. So in the meantime, we want to take this opportunity to appreciate the presenters. They've kept time. They almost went over time during the presentation, but they still have 1 minute remaining at this particular time. I can see, oh yes, there's one more question. All your isolates identified up to genus level, but not to species level. But not to species level. So when you did inhibition tests, how do you know which species is responsible? I think we can handle this as the last question before we move on to the next presenter.
That's a good question. I think given the time frame, we've also done some biochemical tests. Before even going to the molecular level to validate, we've also conducted some biochemical tests to be sure that we have at least significant level of identity associated with the identity of the microorganism that we have indicated in this chart. So in addition to morphological characteristics, we also did biochemical tests. But as I've said, it is an ongoing project. We are going also to validate using the molecular tools.
Okay, thank you so much, Leonard, for that presentation. Thank you, everyone. We have other questions that have been asked. I want to remind each one of us that these questions that are being recorded in our chat, they're also being recorded by our rapporteur, Dr. Caroline Kipkoech, and the presenters will take time after this to also give responses, and these responses will be directed to us because we have your contact. So thank you so much. If we have
Chair.
Yes.
I was just wondering whether Diana is there because she was claiming that she is in.
Yes, she claimed she is in, so I want to confirm.
I'm in, Professor, and I'm ready.
Okay. Thank you so much. Thank you, Prof, for pointing that out. So the next presenter, therefore, will be Diana. You have 10 minutes for the presentation. Again, if you have any questions, please do not hesitate to put them on the chat. At the end of these presentations, we're also going to be doing some surveys, so we'll update you accordingly. So Diana, please continue.
Okay. Good afternoon, everyone. My name is Dana Odubengo. First, I apologize. I had some technical problems. So I'm going to present on effects of intra and intergeneric grafting on physical and biochemical properties for service, physiology, and sensory attributes of tomato fruits. Professor Willis Owino and Dr. Irene Orina are my supervisors. Introduction and statement of problem. Tomato is an important vegetable and it's being used in various houses as a common diet, and it's also a major source of income for small-scale farmers. It has also been known for contributing to different nutrients such as vitamin C, beta-carotene, and carotenoids such as lycopene, which is important in preventing some diseases. So bacterial wilt has been associated to cause close to 100% post-harvest losses, and hence grafting technology being used as a control. So most, from my literature review, most studies tend to focus on controlling the bacterial wilt, but overlook the quality aspects. So there's a contradictory reports on the quality attributes for those few studies that are focused on the quality aspects. So grafting effects on tomato quality is not clear up to date. So that's the problem. And this is just a picture showing the symptoms of bacterial wilt on tomatoes. My objective is to determine the physical, biochemical, physiological, and sensory attributes of grafted tomato of specific maturity stages. So for my case, I'm going to focus on the mature green, tannin, and the red stage in relation to their controls. So my experimental procedures, an F1 is from my study, an F1 is the control. So it's been grafted on African eggplant. So that's intergeneric grafting species from the same family, and also different same family and different genus. And for intergeneric grafting, I grafted an F1 on Japanese tomatoes, which are resistant, have been known to be resistant to bacterial wilt. So this is Armada and BB. The methods, I did weight using a weighing scale, texture using a handheld penetrometer, and the results were expressed in neutrons. Then vitamin C and lycopene, which were analyzed by HPLC, and respiration and ethylene gas production rates, which were determined by the gas chromatography machine. So for sensory evaluation, I used a semi-trained panelist, and the results were analyzed using a 9-point hedonic scale. So this is just a photo showing physical characteristics. As you can see, the Z and F1 control rate, the flesh it's showing, you can just see that they're totally distinguished, and the size also, the control is slightly smaller, but the grafted, which is an F1 grafted on Armada, is slightly.
Observation.
Uh-huh.
My observation.
Yeah. Hello. Hello.
Hello, Diana, that could be a technical issue. The technical team kindly check where this could be coming from.
Mm-hmm. So can I continue?
Yeah, please continue as the technical team checks where the source of the noise is.
Okay. So my results, weight which was expressed in grams, the highest at the mature green was BB, and the highest at the tannin was Armada, and the highest at the ripe stage was Armada too. For all the maturity stages, Sangawiri was the lowest in terms of weight. So, and there was a significant difference between the grafted tomatoes and the control, whereby for all the varieties, whereby for Manire, Armada, and Armada BB, BB and AB2 was significant. There was a significant difference in that they had a higher weights as compared to the control. But for Sangawiri, it was significantly lower from the control. For firmness, the highest was Manire for all the maturity stages, and there was a significant difference between Manire, AB2, Armada, and BB and the control. And so from what I saw, grafting led to fruits with thicker skins and hence farm. That's why there was a higher resistance for the grafted tomatoes. For vitamin C content, for all the maturity stages, Armada and BB had the highest, and there was no significant difference between the Manyire, AB2, and Sangawiri grafted tomatoes and the control. This was the same for lycopene, whereby Armada and BB had the highest, and there was no significant difference between Manyire, AB2, and Sangawiri. For respiration or CO2 production rate, for all the maturity stages, Armada and BB had the lowest, and control had the lowest. So from this study, I saw that grafting increased the quality in that the grafted tomatoes had low rates, which is associated with. Hello.
Mine is observation. Hello.
Oh. Dr. Kahenya, I've been interrupted. So I don't know if you're following.
Okay, Diana, let's sort this. Kindly
hold on a minute, please give us time.
Hold on, yes. Let me call.
Hello, I think we've sorted out. Please continue, Diana. We are sorry.
Okay, thank you so much. So grafting increased the qualities in that for grafted tomatoes, they had low respiration rates, which is usually associated with quality fruits. So ethylene gas production, BB at the mature green, BB had the lowest, and Manyire had the lowest at the tannin, and the BB had the lowest at the ripe stages. Whereas Sangawiri had the highest. Sangawiri had the highest for the ripe stages. Control had the highest for the tannin stage and the mature green stage. So grafting increased the quality for grafted tomatoes. For grafted tomatoes, and hence that's why grafted tomatoes have been seen to have a low ethylene gas production rates. For sensory attributes, it's shown here, as I had said. I was using a 9-point hedonic scale, and grafted is seen to improve quality for sensory attributes of Manyire, AB2, Armada, and BB, whereas there's no significant difference between Sangawiri and the control for all the sensory attributes that were evaluated. So in conclusion, grafting positively impacted on the physical, physiological, and sensory attributes of tomatoes for Manyire, AB2, Armada, and BB. And at the same time, grafting also positively impacted on the biochemical, that's vitamin C and lycopene, quality of tomatoes for Armada and BB, but there was no significant difference between the vitamin C. There's no significant difference in the vitamin C quality, vitamin C and lycopene quality for fruits grafted on Manyire, AB2, Armada, and BB. And it's from the study, it also shows that there's no impact on fruits grafted on Sangawiri rootstocks for all the quality parameters that were evaluated. So in general, intergeneric grafting, which is grafting species from the same family, but different genus, that's BB and Armada. I mean, grafting an F1 on BB and Armada recorded the best quality traits over intergeneric grafting, which is grafting species from the same genus and the same family. And for my case, grafting an F1 on Manyire, AB2, Armada, Manyire, AB2, and Sangawiri. So thank you so much for your time. And always remember, quality comes before quantity. Thank you so much.
Thank you so much, Diana, for the very clear and presentation. Indeed, one of the members of the audience has appreciated the clarity and educative nature of the presentation. So again, members, I want to remind you, if you have any question for Diana, you can put it up on the chat and we'll pose it to her. So Diana, one of the key findings that you said there are some parameters that have been affected by grafting. You're calling it intergeneric grafting, yes? Yeah. And some attributes were not affected. What would be the explanation for that difference or that disparity?
Okay, yeah, the explanation for that is while grafting, you always have to look on the rootstock, scion rootstock combination. So I think it depends on the scion and the rootstock combination.
Okay, thank you for that. So anyone else with a question for Diana, please put it up so that we can ask the question to her. In the meantime, thank you so much, Diana, for such a clear presentation, very educative and very informative.
Thank you.
Yes, so if there are no other questions, then we could then go to the next presenter. The next presenter is, I believe, Clement Komu, working together with Monica Mburu, Daniel Joroge, and Richard Koskei. And Clement is supposed to be, or is presenting to us, the physicochemical, his title is Physicochemical Profile of chia seeds, essential oils, Salvia hispanica L from different agroecological zones in Kenya. So Clement, if you are with us, kindly, the floor is yours. Clement? If Clement is not available, is there any other presenter who is available who can handle the presentation? If you are there, kindly show us by raising up your hand. If not, then we're going to invite the next presenter. I believe the next presenter, I can see he's online. This is Elias Ngodo. Elias working together with Abanas Mutwiwa and Kipchumba Cherono. The title of the presentation is Evaluation of the Effects of Zero Energy Cooler on Tomato Fruits. So Elias, you can start the presentation now. Elias, do you need assistance? Elias, can you hear me? Caroline?
It seems that Sharon is ready. So we might move to the next presenter as Elias prepare, because I can see Sharon already lifted her hand up that she is ready.
Okay.
So you can let Elias sort out whatever problem he's facing.
Okay. Okay, thank you. Then so Sharon is supposed to be on the next presentation that's not timed.
Yes, that has not time.
Okay.
Elias? And then Elias, if he has this presentation, he can kindly forward it to us so that we can be able to share it with him, for him during the presentation if he has difficulties.
Okay, I hope Elias, you can hear us. So in the meantime, therefore, we'll go to the next presenter. The next presenter, this should be the team comprising of Victor Chege, Martin Kimani, Sharon Cheboy, Emily Kendi, Gikunda, Paul Jogo, and Catherine Gamau. The presentation is titled Assessment of Different Ratios of Water Hyacinth Oropit and cocoa peat as propagation media on growth of tomato seedlings. We have a lot of work on tomatoes going on today. So Sharon, you're welcome.
Elias, please stop sharing.
Yes, hi everyone. I hope you're having an amazing afternoon. I think I need Elias to stop sharing his screen.
He has, so you're welcome.
Okay, please let me know if you can see my screen. Can you
Yes,
all
we can
see my
it.
screen?
Yeah, we can see it, but make it full.
Is that okay right now?
Yes.
Okay, hi, good afternoon, everyone. My name is Sharon Cheboy, and I'll be presenting on behalf of my co-researchers, Victor Chege, Martin Kimani, Emily Kendi, and our advisors, Dr. Catherine Gamau and Paul Njogu, who are both affiliated to Jomo Kenyatta University. And we took a research around the topic of assessing different ratios of water hyacinth peat and cocoa peat as a propagation media for the growth of tomato seedlings. And as we all know that water hyacinth is a weed that is native to South America, and it's spread to the other parts of the world as an ornamental plant. And in Africa, it is present in River Kagera, Pangani, River Nile, and locally it has been a terrible menace in Lake Victoria, and it has been reported to interfere with transport systems and aquatic life, whereby we find fish floating in the morning because of oxygen deficiencies. And recently there have been cases of reporting increased cases of malaria and alongside the Lake Victoria. And previous uses and utilizations of water hyacinth has been, for instance, the use of water hyacinth to develop biofuels in a recent Dunga Beach project, and locally women use it to create some sisal-based baskets and also paper and livestock feeds. So the whole idea of this project is to explore different agricultural avenues to utilize this water hyacinth weed that has been named as the most dangerous aquatic weed. So in Kenya, in 2015, there was a conjunction with the World Bank and the government of Kenya to procure a water hyacinth harvester to mechanically control water hyacinth. While we would like to acknowledge that only mechanical control of water hyacinth would not be efficient in controlling the entire lake, it was one of the methods of trying to eradicate and to minimize the spread, which has covered about 4,000 meters square of the lake. However, this type of control was futile, since not sooner had it started that the machine broke down and the government cited mechanical challenges and high maintenance costs for the machine. And I've had recent talks as early as February last month that they are trying to fix it, and let's just see how that goes. So locally, cocoa peat is one of the most commonly used soil propagation medias in nurseries. And what we would like to acknowledge is that cocoa peat does not have any nutrients, but water hyacinth is highly nutritious and it has NPK, which is also absent in cocoa peat. While one of the challenges that local growers face is that most of the cocoa peat is largely imported, and water hyacinth peat could provide an alternative or a substitute to such media. So the objective of this project is to determine how efficient water hyacinth peat and cocoa peat ratios would be as a propagation media for tomato seedling, and to determine the chemical properties that this water hyacinth peat that we would make would possess, and also assess some of the growth parameters in tomato seedlings that have been grown from this water hyacinth peat. So the research was carried out in JKUAT with the support of the JKUAT nursery area, and we used tomato seedlings as the test crop, and water hyacinth was harvested from around Jomo Kenyatta University, which is sad that we already have water hyacinth in the university, around the university. So this is the visual representation of the activities that were carried out. We mechanically cut water hyacinth into smaller pieces and fed it into a biogas digester just to produce the peat. And this is a visual representation of the ongoing decomposition. And it took about 90 days to create a well-decomposed peat, but then it was a very cold part of the year, so it could be shorter. My assumption is that it could be shorter if it is in the hotter part of the month. So after the decomposition process, it was dried and taken into the lab for assessment. And after that assessment, it was crushed into much smaller pieces and mixed into the ratios that I will show you in a bit. And the tomato seedlings were planted. So in the laboratory, we found that potassium was sufficient. Both potassium, nitrogen, and phosphorus was sufficient. And this is the visual representation of the methods, of the laboratory methods that we undertook to ensure this. However, the pH was slightly on the higher side at 8.3, and our average expectation was that tomatoes would thrive in a pH of about 6.2. And the EC was also slightly high, but then it was also in line with previous research. They also found that the EC was high, attributing it to a bit of salinity within the water bodies and whatnot. So to work around the high EC, we rinsed our media in and we rinsed our media and brought it down to 0.7, which would be ideally perfect for the tomato seedlings. So this was an experimental layout, and our media was mixed in different ratios of 50% water hyacinth and cocoa peat, 100% water hyacinth, 25% water hyacinth and cocoa peat, 75% water hyacinth and cocoa peat, and 50-50 each. So we carried out parameters. Two of the parameters that we carried out was plant height and root length. And this just shows the visual representation of the significance that our media had on plant height and also root length, because we saw the disparities in the root length for each treatment. So as you can see that the longest roots that we found was in 75% water hyacinth and 25% cocoa peat, and the shortest ones was in 100% cocoa peat, which is the last graph at that, which is the last bar in the graph. And also the plant height was a bit different, with the longest tomato being 25% water hyacinth and the shortest being 100% water hyacinth. So this is the visual representation again of the general presentation of the samples that we selected to represent each treatment. As you can see, the fourth plant from my left or from your left is 75% water hyacinth, and it is a very dense root compared to, for instance, 50% and 50% cocoa peat. So we also tried to work around the issue of anchorage and to test whether our media would be able to hold water and provide support into the plant. As you can see, the very last, this 25% water hyacinth is a fairly healthy plant, and it's a very sturdy media. And unlike the 100% cocoa peat, which was borderline breaking down, and also the 50%, but then generally, all of these represent a fairly steady anchorage. So yeah, so some of the conclusions that we drew is that 25% water hyacinth, a ratio of 25% water hyacinth and 75% cocoa peat showed us the highest emergency percentage at 100%. So this would be ideally what we would recommend. It also gave us the highest and the densest root. However, 75% and 25% cocoa peat was the lowest. It had an emergency percentage of 93%, but had the longest roots. And we also attribute this as to the high NPK as the content of water hyacinth increases. So it has that impact on growth parameters. While the pH was slightly high, we had to adjust that and our adjustment of choice was vinegar. And yeah, we brought that down to about 6.2. So to conclude the entire research, we concluded that water hyacinth peat is actually an efficient growth media and when we mixed it in different ratios of water hyacinth, the complementing of the physical and nutritional properties present in water hyacinth and they would support root growth adequately. And 25% water hyacinth was one of the most best performing, even the visual representation of it. And this is one that we would recommend just from this brief research. So some of the gaps that we would ideally recommend is that we didn't analyze the possibility of heavy metals in water bodies because we understand that could be a problem, especially if we are getting into the fruiting stage. So this could be an ideal gap that people or future research could take up. And also the economic scalability, because our research was at a smaller scale and it was limited by time. So we didn't get to explore the economic aspects or the economic practicalities.
Please conclude. The time is up.
Okay, I am finished. I am complete. I have completed, and we just want to acknowledge the support of the Department of Horticulture and IEET and the manufacturing chair, and also the intellectual support of our advisors, Dr. Catherine Ngamau and Paul Njogu. Thank you.
Okay, thank you. Thank you so much. Thank you so much for a very clear presentation, very informative again, high quality presentation from you. We, however, have one or two questions for you. And the first one would be, what is your conclusion on the use of 100% water hyacinth media or media made out of 100% of the water hyacinth?
Okay, so yes, I can hear you. Our conclusion was that you could actually use that 100% as a media on its own, but comparatively with how it generally performed based on the parameters that we assessed, a balance with cocoa peat would produce a better yielding seedling, but it would still be an efficient media because the emergency percentage was still high and the parameters were still comparatively on an average scale compared to the other mixtures.
Okay, thank you. Thank you so much. There's also the observation that from your presentation, from the graphs that you presented, the root length with 75% water hyacinth and 50% water hyacinth seems to be fairly the same. Looking at the error bars, so the question is, what would your comment be on using 75% and 50%?
I'm sorry, I didn't get your question. Could you please clarify?
The observation is, if you look at your the graph that you presented, you look at the root length graph, and you look at what the bar for 75% water hyacinth and the bar for 50% water hyacinth, taking into consideration the error bars, which shows the real or the significant difference between the two, then would you then conclude that there are no significant difference between the two, considering that they look barely, according to the observation, they look the same, or they basically show that there might be no significant differences?
Yes, I understand you now. What I would say is that while we were carrying out the experiment, we found that we had quite a bit of challenge with handling the texture of the 100% water hyacinth. So what we would recommend is that we can't categorically place or pinpoint any reason that that difference could be, that was not significantly different, but then it's also a very slight difference.
So thank you so much, Sharon, for the very good presentation. Again, it's appreciated even by other members of this team that you've done some very good work and it's very informative. So we want to stop there for now. We appreciate that you've done some very good work and we want to go to the next presenter. And I don't know whether Elias, you've been able to solve the problem that you had before. Elias? So you can stop presenting, Sharon.
Sorry, where do I click to stop presenting?
Oh, then if Jeremiah here, stop the presentation.
Okay, thank you, please.
Elias? Elias, I can see you online, but are you able to hear us? You could write something on the chat if you have any comments, Elias. If not, then I'll call upon the group comprising of Jacinta Muiruri, Frida Wanzala, Mwajita, Mwashasha, and Agnes Kavo to present on the abundance and diversity of arbuscular mycorrhizal fungal AMF spores isolated from the rhizosphere of different cropping systems in central Kenya. So I believe, Jacinta, you're there?
Hello, everyone.
Hello.
Can you see my screen?
Yes.
Yes.
Thank you very much. Now, I'm Jacinta Muiruri on behalf of my supervisors, Dr. Frida, Dr. Mwashasha, and Dr. Agnes Kavo. The title of this presentation is The Abundance and Diversity of Arbuscular Mycorrhizal Fungal, the AMF spores, isolated from the rhizosphere of different cropping systems in central Kenya. Now, in the introductory part, mycorrhizae is the heuristic symbiotic association between soil-borne fungi and plants, whereby the mycorrhiza benefits from the sugars and carbohydrates that are produced during photosynthesis, and the plants benefit greatly through the nutrition that is obtained through the mycorrhiza, and also through the nutrients such as phosphorus, potassium, zinc, and nitrogen, among others. The AMF, they are important in majority of cultivated species of plants during the colonization of the roots, and they also support the plant growth during adverse climatic conditions. These adverse climatic conditions include among them the stresses such as water stress, which most of us refer to as drought, and also salt stress, and also protect them against soil pathogens. For example, tomato is protected against Phytophthora parasitica, while potatoes are protected against Fusarium subcutaneum, which is also called the dry rot tubers. Plants also benefit, as I said earlier, from the nutrients, and this is because of the extra radical mycelium that explores a greater volume of soil and also translocates nutrients from soil to plants. This overall improves the plant nutrition. Some of the problems that are being faced is that the high diversity of AMF that is observed around the rhizosphere has not been explored. So we are not sure about the abundance of the AMF in the roots of these plants. Considering the importance of these spores in the plant roots, it is important to know the spores in numbers and also the types that are there in these spores. And that is why we are doing the characterization. We did also the characterization of the rhizospheric AMF. Another problem faced is that most seedlings are affected during acclimatization, so they are not able to adapt to the harsh conditions that they find in their new environment. And use of AMF, on the other hand, ensures that these plants tolerate the hardship of the area and they're able to adapt well. Another problem we are facing is that due to the industrial elements, also the overcultivation and undercultivation of lands, most of these mycorrhizae fungi has been exhausted. So you realize that most farmers end up using the synthetic fertilizers, and most of the effects of these fertilizers are documented. We also experience them, like the emissions, the problems we have of the pollution. So these problems can be addressed by identifying the spores that are available in the soil, so that we are able to either multiply and introduce them to soils without use of the inorganic fertilizers. The objective of this study was to determine the abundance and diversity of the AMF spores that were isolated from the rhizosphere of different cropping systems in Kenya. In the methodology, we obtained soil from the rhizosphere of 3 plants, that is papaya, banana, and grass, and these were obtained from Mwea, Mitungu, and JKUAT at a depth of between 0 to 20 centimeters. It depended with where the soil auger could reach, but any measurements in between 0 to 20 centimeters, you obtained that soil. This soil was then mixed with water, and it was thoroughly mixed and left to decant for about 30 seconds, and was then washed through the 250, 100, and the 45 micrometer pore sieves. The final sieve of the 45, those contents were backwashed into a beaker and then swirled, and then these contents were then decanted into the 50 ml centrifuge tubes. And because before you go to the centrifuge, you have to balance them by weight, and they were centrifuged for 5 minutes at 1,750 revolutions per minute. After that, the supertannant was poured, and 48% sucrose solution. This 48% sucrose solution consisted of 227 grams of sucrose dissolved in 500 ml of water. They were added now and balanced again by weight and centrifuged again, now with the sucrose solution for 15 seconds at 1,750 revolutions per minute. The supernatant sucrose was emptied through a 45 micromesh sieve, and these were then transferred to a gridded Petri dish for counting. And this is where now abundance of spores was recorded and also characterized. This methodology was repeated 27 times to represent the 3 plants, the 3 locations, and the 3 replications. In our results, it is clear that the spores obtained from grass that was collected from Mitungu were the highest, and this is now the average mean spore for per the 50 grams of the sampled soil, and this was followed again by Mitungu banana, Mitungu, the Juja grass, the grass, and also the Mwea banana among, and the results are clear in the graph where we have the Juja grass of having the most spores, and this clearly shows that there is a great difference in the amount of spores that were isolated from these areas and also the plants. In the characterization, this was done morphologically, and it was based on the color, the shape and size of the spore, and they were distinguished according to the descriptions provided by the International Culture Collection of Abusula and Vesicula Abusula mycorrhizal fungi, that is the INVAM. And among the spores that were isolated, the 54 of them, I didn't represent all of them, but a few were represented just to appreciate how all the 54 spores were recorded. And this was, again, as I said, according to the shape of the spore, the color, the Melzer's reaction. Melzer's reaction there is a Melzer's reagent that is added on the spore when it is on the slide. And once it is added, it distinguishes the spores further from its color. It distinguishes further by addition of the Melzer's reaction. We also took the diameter of the spore in micrometer. The spore surface, just the appearance of the spore surface, and we were able to categorize these spores morphologically to their families, genus, and species. So I only sorted the 9 spores to represent every area and a plant to appreciate the results of this study. The colors ranged, and you can see that after adding Melzer's reaction, there is a difference in all of the spores. Some were reacting, some did not react, and this further gave us the taxonomy to be able to differentiate the species, for the species that were able to be isolated and identified. For the isolated species, the family's distribution were as follows. We had the Acrosphoraceae family giving the highest number, and these were followed by the Glomeraceae family, the Diasporaceae family with 10, while the Diversisporaceae family had the least number of genus and species. To be able to discuss the results, we can say that the AMF, they are strongly reliant on their host plants and have different levels of specific host pairing. So that we can also see that the variations in the number of spores recorded among the plants and the different areas. So it means that with different plants, with different areas, there is a variation of the spores in their genus, in their species, and also the abundance. And this abundance could be due to abiotic factors, and especially the differences in the temperature and the rainfall amounts in the area.
Jacinta, kindly plan to conclude.
Okay. Because these are now the different temperature and the rainfall for the 3 areas, for the Mwea, for the Mitungu, and for the Juja. It is clear that the 2 families of Acrosphoraceae and Glomeraceae dominated the 3 areas. So color is a big aspect, although this depends on the maturity. The older they get, the colors change. And also addition of Melzer's is also a good indicator of the genus and the species. To conclude, it is clear that the abundance of AMF from the 3 areas, from the 3 plants are different. And this great diversity of the AMF is important so that we can be able to know whether spores can be added or not added. We can be able to isolate more spores and add more spores to these plants so that we can improve on their growth. And we can also know that the cropping systems and the associated plants contribute greatly in the differences of the genus and the species of the AMF. Thank you, everybody who has participated in this.
Thank you so much, Jacinta, for that very clear presentation. Highly appreciated, a lot of things to learn for some of us. So I want to see if there are any questions. In the meantime, Jacinta.
Yes.
As you concluded, you explained the importance of these spores or the diversity of these spores.
Yes.
Now, to a layman,
yes,
what would you tell them is ideal? What would be the ideal mixture of these spores? Because I saw they are diverse. What would be the most ideal? What would be the perfect situation for us to get the best yields?
Now, what happens is that to isolate a single spore really takes a lot of time. So what you do is that once you isolate the single spore, you can multiply it. There are plants that are called trap catchers. And these trap catchers, even if you put one spore, within 2 or 3 months, they multiply very fast. So all you need to use are the roots. And it does not matter how many or how the amount of spores in the roots, the more the better, because you see they have diverse uses. So you can use them for to extract nutrients from further areas of the soil. So the more they are, the better to the farmer. But because they are okay, you only get a few of them when you're obtaining them. So when you multiply through the trap catchers, just use the roots of the trap catchers to introduce those plants.
So if I'm getting you right, very soon we can be buying these spores and adding them into our soils and improving the productivity of our farms.
Yes, and you don't use fertilizer at all, and you'll be surprised on the yield.
Thank you for that. Any other questions for Jacinta? So in the meantime, Elias seems to be experiencing challenges with his computer speakers. He can hear us, but we can't hear him. I will, as we wait for any other question that might be posed to Jacinta, we'll give him probably an opportunity to see whether he can get an alternative computer. If he's around Jomo Kenyatta, I think we can easily be able to help him. If it's outside of the premises, it might be more difficult. So Elias, kindly update us if you still can hear me. So in the meantime, we've had an opportunity to listen to 5 different presentations. The first presentation was by Agnes and team, and they talked about the farmer knowledge and preference for orange flesh sweet potato production and utilization in Isiolo County. And from that presentation, we were able to get to understand the changes that are occurring in different parts of our country. They informed us that this is formerly a nomadic pastoralist community, and they are now adopting the cultivation of sweet potatoes. We also got to understand that they also have preferences. They've grown to prefer certain varieties of the sweet potatoes. The second presentation was by Kireka and team, and they talked about the isolation and characterization of rhizospheric microorganisms from bacterial wilts, endemic areas in Kenya. I think the title changed a little bit during the presentation. And from them, we were also able to understand, or we were able to get to understand the relationship between the rhizospheric microorganisms and the control of pests and diseases in tomatoes. The third presentation was by Diana, who also talked about grafting of tomatoes and showed the advantages of intra and intergeneric grafting on the physical and biochemical properties for surface physiology and sensory attributes of the tomato fruit. And we also had an opportunity to hear from Sharon on the assessment of the different ratios of water hyacinth in production of propagation media for growth of tomato seedlings, which also saw is actually very possible because they're able to get some very good media for the production of the seedlings. And finally, we've heard from Jacinta on the abundance and diversity of avascular mycorrhizae fungal spores isolated from the rhizosphere of different cropping systems in Kenya. And we've concluded by saying that if we could get more of these, we'd be able to improve yields if we could isolate more of these. So I don't seem to be getting any response from Elias. And so I want to invite all of us to make sure that we feel there is a link that has been shared by the rapporteur, Our rapporteur for this session has been Dr. Caroline, and she's shared with us a link several times. If you look at the chat, it's a simple Google document. So you just click on it, and it's within a very short time, you have filled in a lot of details, and you could then be able to give us a response of some feedback about this session. I want to thank each one of you. I think we've listened to all the presenters, with the exception of Clement, who was not available, and Elias, who's faced some technical problems. I would want to appreciate each one of you for taking time to attend these presentations. It would have been very unfortunate if the presenters were to present to 2 or 3 people, but the fact that we started at around 30 of us and we've ended with around 30 of us means that the presentations have been very interesting, have been very informative. The rapporteur has taken note of all the responses that have been taken. We are, like I said earlier, we are recording anything that you write on the chat is being recorded for future reference. And these questions, if there's any question that was asked and has not been responded to, it will still be responded in the proceedings that we write out of this workshop. So from me is to thank you for your participation. I want to invite Caroline for any comments on this. Caroline?
Yes, Chair. Thank you so much for taking us through this session very smoothly and very nicely. Thank you for all the presenters, and thank you for everyone who participated. We still have 2 more sessions tomorrow. Please join us in agriculture so that we finish and learn more and pick some important information. Thank you so much. God bless you. Have a good evening. So we are through now with our presentation. You're free to leave. You can join the exhibition, but the other sessions have ended. I think we were the last group. So you can have a good evening as you go home. See you tomorrow. Thank you so much.
Hello, can you hear me? Something maybe before I forgot. To all participants, to all presenters, if you have not shared your presentation with us, please do, because we are experiencing, some of the presenters are experiencing technical hitch from their end. But when you have that presentation, you can share from our end. Please share with us your presentation if you have not done, you are presenting tomorrow.